So now I'm very excited to introduce the last speaker of the conference. So Dr. Connor Bevington is a postdoctoral research fellow at the UBC PET MRI Imaging Center at the Department of Physics and Astronomy at the University of British Columbia. And today he's going to present his work on pattern analysis of PET MR data enabled by advanced MR denoising, application to exercise impacts on brain energetics in Parkinson's disease.
Perfect, thank you. Yeah, so my background is on PET mostly, where quantitation is kind of the norm. So we're very excited at our group to be now merging kind of our analysis and processing tools with a joint data set, PET and MRI at the same time. This application is for brain energetics, which refers to the production and utilization of energy in the brain. And for this, we need to quantify metabolic rate of oxygen and metabolic rate of glucose at the same time. If the ratio of oxygen to glucose is 6, then we have fully aerobic energy production, which is quite efficient. And if this ratio dips below 6, then we have some degree of aerobic glycolysis, that is, using glycolysis despite the presence of oxygen. And this has been mapped out in the healthy brain before by Vaishnavi and al., and you can see there are regional differences across the brain in terms of aerobic glycolysis. In PD, there are well-known alterations to metabolic rate of glucose, regions of hypermetabolism and hypometabolism. And similar alterations to blood flow and oxygen metabolism have also been observed in these regions. However, there is a hypothesis that mitochondrial dysfunction is present in PD, and this would mean that there's a possibility of increased aerobic glycolysis to meet these energy demands where the mitochondria are not working properly. So this would imply that the alterations to glucose and oxygen would be discordant in such affected regions by mitochondrial dysfunction. And furthermore, there's been some preclinical research that indicates exercise actually helps improve mitochondrial function, mostly in rats. However, PD-related mitochondrial dysfunction has not been characterized, and therefore any effects. Work is to characterize alterations to brain energetics in vivo in PD, and then to see if there's a positive effect of exercise. Okay, methods. So to quantify these both at the same time, this is where the MRI comes in. So for oxygen, we're using a dual-calibrated bold fMRI technique. The operational equation for oxygenation is here. So we need to quantify blood flow, the oxygen extraction fraction, and then the arterial oxygenation. So for this, we're using a dual echo PCASA sequence, and this is acquired during hypercapnia and hyperoxia gas challenges. The first echo is an ASL sequence, so we're getting CBF from there. Echo 2 is bold signal. While this is going on, we're simultaneously measuring the end-tidal partial pressures of the subject, what they breathe out. And then from the oxygen end-tidals, we get the arterial oxygenation. And then from a biophysical model that incorporates the bold signal, the blood flow signal, and the oxygenation, we can get the oxygen extraction fraction. And on the PET side, we're using FDG to get metabolic rate of glucose. These are both acquired, as I mentioned, in a single scanning session on a simultaneous PET MR scanner, the GE Signa. One problem with this technique on the dual calibrated bolt side is that the data are exceptionally noisy. So going back to our operational equation here, if we go through step by step all the ingredients, the blood flow images are generally quite robust. The arterial oxygenation is a global constant and is also robust. So this leaves the oxygen extraction fraction. It's a heavily derived parameter with a number of inputs, so it's highly sensitive to noise in these measured values. And just for reference, here's what a single subject's data may look like. Normally in healthy subjects, you have uniform OEF across the brain at around 0.4. You can see there's a number of non-physical values that have been thresholded out here. However, when you average over a large number of subjects, you get a more realistic map kind of lying in that 4 to 4 range and looking relatively uniform. So the takeaway is that we don't really have robust estimation on a single subject level. So our solution is to use some MRI denoising techniques that have been informed by kind of our PET expertise in terms of denoising data, and we employ a lower dimensional feature space to guide the denoising. To construct the feature space, we go to the standard Davis model of the bold signal. You don't need to know all these terms here. The important thing is that there are 2 major sources of variation in this equation. The first one is the scaling factor M here, and the second one is the deoxyhemoglobin concentration at baseline, which is proportional to OEF. So our denoising process works as follows. We first map the original data to a feature space. The first dimension of this feature space is sensitive to M, the second one sensitive to OEF. And then in this feature space, we perform k-means clustering to identify voxels with similar physiology. And then we can average over these back in the original data space, the bolder the ASL signal, to create a set of basis functions. And then we can reconstruct a denoised version of the data where each voxel signal is now a weighted sum of these basis functions. And to inform how these weights are constructed, we go back to the feature space and then see how far away each individual voxel is from its K-cluster centroid. And finally, just to ensure some consistency with the original data, we apply a data consistency correction, which is known as the hyper operator. Subjects for our work so far, we've scanned and obtained CMRO2 and CMRglucose from 12 non-exercising PDs. Here are their demographics below. And they're scanned before and after 6 months of various exercise intensity. Right now, we're still waiting for 2 subjects to complete their follow-up. They're currently in the study, so 10 have completed for now. And also, we've measured the motor disease severity, which for Parkinson's, you can use a scale called UPDRS, and the aerobic fitness level is measured by VO2 max, and this is done before and after as well. So going back to what we're looking at in terms of aerobic glycolysis and mitochondrial function, we need a metric that combines the 2 data that's sensitive to aerobic glycolysis. So we use a joint analysis technique that's been adapted from the literature. And basically what we do is for each subject, we plot the regional glucose on this axis and the regional oxygen on this axis. And then we perform a 45-degree rotation of the data. So we have 2 new sets of orthogonal axes. This is kind of showing the general energy production. If you're up here, you're using a lot of oxygen and glucose, so high energy production. But if you look at the orthogonal axis here, which we call relative cost, if you lie in this upper plane right here, this is where you have relatively high levels of aerobic glycolysis because the relative glucose consumption exceeds that of the relative oxygen consumption. So relative cost is a measure that is sensitive to aerobic glycolysis. To analyze aerobic glycolysis in vivo, we're using a technique called partial least squares. It's a form of pattern analysis. So what PLS does is it identifies brain behavior interactions, and it does this by analyzing the covariance between a set of imaging data and clinical variables. So as I mentioned, we're going to be using relative cost to try to look at aerobic glycolysis. So this constitutes our imaging data. And then our clinical data in this case is the motor disease severity metric and the aerobic fitness metric as well, which is also for each subject. PLS works as follows. You first just do a data centering and normalization step for each matrix, and then you compute the correlations between the 2, and then you perform singular value decomposition on this correlation matrix right here. And what this allows you to do is to extract clinical patterns right here and spatial patterns on the other side, so either side of the decomposition. The subject scores, which are detailing how much each individual subject expressed these patterns, are constructed by projecting the original data, so the imaging data onto the imaging patterns, and the clinical data onto the clinical patterns. And as a result of this decomposition, what ends up happening is you get a maximal covariance between these 2 sets of subject scores for each pattern, and also so the different sets of patterns. Results. First, going back to our denoising step that we were mentioning, the OEF maps that we get are more accurate and reliable after denoising. So first, we started with some simulations. So we had a uniform background region with a small lesion that's a lower OEF. And you can see with some standard denoising, what you get are pretty much what I showed earlier with the real human data, a lot of non-physical values. If you really get aggressive with your filtering, you can denoise further, but then you lose the contrast of this lesion, whereas our proposed denoising method is able to preserve that background while also preserving the lesion. And we also see similar results in real healthy human data. Again, for healthy data, you'd expect it to be uniform. Some subjects' data aren't as noisy, so there would be minimal difference between the 2 methods. But for a number of subjects, the noise is quite high, leading to, again, a lot of non-physical values, whereas our method is able to correct for this. And then on to the pattern analysis PLS results. What we do find are abnormal energetics PD that are modulated by aerobic fitness. So I'll step through this a bit slower. On the left-hand side, you're seeing the pattern. And just for simplicity, I'm only displaying regions that fell out of the pattern that are known to be affected in PD and have abnormal energetics. So this would be where the relative cost or the aerobic glycolysis is elevated. So we see occipital parietal regions with elevated cost, as well as in the substantial nigra. In terms of the pattern expression, so this is how much each individual subject expresses the pattern. What we see is if you analyze this as a function of motor disease severity, you can see the pattern expression increases, while simultaneously it decreases with aerobic fitness. Now, the one wrinkle in this is that we only see these correlations post-exercise. So to try to understand this, I'll just flash the entire pattern right here, shown in a bar graph. And then on the left-hand side right here, you see the expression of this pattern for motor disease severity before exercise and aerobic fitness before exercise and then after. So those correlation plots you saw in the previous slide are described here, again, going up with motor disease severity and going down with VO2 max or aerobic fitness only post-exercise. So what this tells us is that this is not a disease progression pattern because we would have seen that pre-exercise as well. What we're thinking is that pre-exercise, all the variance that you're really seeing in VO2 max is going to be dominated by the subject's genetic predisposition because all of these PD subjects were not exercising. Whereas after exercise, more of the variance you see in VO2 max is going to be sensitive to how well they did during the exercise study, so how much they improved their fitness. But what we're seeing is that these improvements in brain energetics are constrained by the disease severity. So that means if you're quite far along in the disease, your ability to modulate the exercise is constrained. So we really only see this interaction happening post-exercise because beforehand, none of the subjects were exercising. So to conclude, we found that aerobic exercise modulates aspects of brain energetics in PD. This supports some of the preclinical research I mentioned at the beginning and just general clinical observations in PD. Disease progression seems to be attenuated, or exercise, in other words, is a neuroprotective mechanism for PD. Our results suggest that intervening earlier in disease is better just from that interaction. If you're not as far into the disease or your motor severity isn't as extreme, then it seems that you can have greater improvements in brain energetics by exercising. These results were only enabled by advanced MRI denoising to make these fully quantitative and reliable on the subject level. And it also further demonstrates the utility of using a joint pattern analysis technique on quantitative PET and MRI data sets. And it might provide a possible framework for investigating brain energetics and other diseases or other interventions within PD. Future work, so we want to check whether exercise modulates other aspects of pathology in PD. For these subjects, we've also obtained resonance data from MRI, DTI data, and SWI data as well. So we can get QSM data as well and look at what the other talkers or speakers today have been talking about iron deposition to see if there's any modulation there by exercise as well. And I'd just like to thank our incredible team who's made all of this research possible. Thank you.
Thank you very much for your presentation. It was very interesting. Mark, do we have any questions?
Yeah, we do. Olivia Perron asks, how long is the P-Castle-Bold acquisition?
It has to be long for this one. It's a 21-minute sequence. The bouts we do of CO2 are 2 minutes in duration. The ramp up's quite quick for CO2, just a couple of breaths. And that lasts 2 minutes, as I mentioned, and we repeat it again later in the scan. And then for oxygen, we do a 4-minute one. So when you have all those interventions followed by a little baseline period for them to kind of catch their breath, it ends up being, 21 minutes long.
Okay. I'm also wondering, you mentioned that some subjects are noisier. Is that like behavior or anatomy driven?
Yeah, so what we find, we have a targeting, so basically the signal you're looking for is the change in Parkinson, right? The change in the bold signal, and it's kind of brain-wide influenced by mostly CO2. That's the stronger signal change you can get. You get a small signal change from increasing oxygenation as well. But during that, we're targeting, if this means anything, +8 millimeters of mercury for the partial pressure of CO2. And for some subjects, the older the subject is. And also, there's an effect of if you're more fit, you actually need more absolute concentration of CO2 to cause these brain changes we're showing, which can be quite uncomfortable for the subject. So some of them, we had to dial it back a little bit. And in that case, we see a smaller change in the brain as well. Yeah, you're usually looking at changes of the bold signal of about 2%. So with the high noise already in these signals, that can be difficult to distinguish.
Okay. All right. Thank you. Dr. Christopher Rowley asks, he says, great talk. And then he says, in terms of analyzing the VO2 max and inferring activity levels, did you have participants wear an activity tracker or do a questionnaire? The VO2. Sorry. Okay, I'll finish the question. The VO2 max range you were showing could also be impacted by walking intensity and frequency for pre-exercise values.
Yeah. So beforehand, the way we ensured they were they were non-exercising was not through the VO2 metric itself. Like we did see some who technically were non-exercising but had relatively high VO2 maxes, and I think that's probably a combination of maybe in their younger years doing it, and also they mentioned genetic predisposition. During the intervention itself, they are wearing a fitness watch and a chest heart rate strap, so that's for all their high-intensity exercise techniques. We've stratified this. I didn't mention in the talk. There were 7 of those subjects who actually did a very stringent supervised exercise protocol, and the others were just following over time. So that's how we kind of get the range of changing in VO2 max. The subjects who are just walking or keeping up their day-to-day non-exercising techniques. So when I say non-exercising, I mean not going on a bike or going for a run or something that's really going to improve your VO2 max. For those subjects, we didn't see as big of a change in VO2 max or even a negative change. And then for the subjects who did the intense exercise one, where we were tracking it and had a trainer delivering these 3 times a week, we saw on average an increase of 30%. So that's where that spread of VO2 max is coming from. And we believe because we were tracking it quite strongly, then we could maybe use those metrics further down to show like total activity load or things like that, not just a metric of VO2 max.
Okay. All right. Thank you. As a final question from Dr. Millard Chakravarty, he says, how generalizable do you think these results are given the sample size and the analytical methods? Yeah,
so it's a small sample, obviously. In terms of PLS, like the way you get around generalizability as well, to say this is not just a pattern inferred from this subject set, is you do permutation tests as well. So that's the generalizability of the pattern on itself. So that passed a generalizability test here. And then there's also a bootstrapping procedure as well to identify that these regions are actually contributing to the subject or the pattern as a whole. And that's done by kind of swapping out different subjects. And in that case, you're making sure it's not just 1 or 2 subjects that are driving the pattern. It's kind of all of them are consistently seeing it. In some regions, there were that didn't pass this reliability, so I didn't show those. But that would be a case where it's present in one subject, but not really all the other ones. So I think they're fairly generalizable, but due to the small subject size, we'd have to probably expand it. And we do have a future study that's not only doing exercise, but things like diet and mindfulness as well. So I think we're going to try to get this exact same protocol loaded into their study as well, and to see if it expands in a larger subset. Because we're quite limited in Vancouver, at least, finding PD subjects who don't exercise, because the norm now is when you get a diagnosis, is your physician will say, start exercising, and Vancouver is quite an active place as well. So we had a really, really tough time getting subjects who are non-exercising into our study.
A good problem, I suppose. Thank you very much. Super interesting stuff.
Thank you.